Hello, everyone, and welcome to the webinar. Today we're going to be doing a webinar called Searching for a Cure, the ROS1 Cancer Model Project. And it will be presented by the ROS1ers with grateful assistance from Longevity and from the University of Colorado. I will share my screen and get started. We're going to have three speakers today. Myself, I'm the co-founder of the ROS1ers and the board chair, also a patient research advocate, Janet Freeman-Daly. On Lee from the University of Colorado, he's a senior professional research assistant and the manager of the Doebele Lab. And Dr. Amy Moore of Longevity, who's their VP of Global Engagement and Patient Partnerships. To get started, we have a video that shows what a cell line looks like and what it's about so that we can start talking about cancer models.
What is a cancer model? Basically, a cancer model allows researchers to study live cancer cells outside the human body. One type of model is called a cell line, which consists of immortalized cancer cells growing in a lab dish. Some other model types are mice, genetically engineered to have cancer, or that have human cancer cells growing under their skin. Researchers use these models to study cancer biology and develop new treatments in the laboratory. Today, we're going to focus on cell lines derived from patient tumor cells by touring a lab at the University of Colorado that makes and studies cell lines.
So here is our tissue culture room, and what we have here are tissue culture incubators where we maintain live cells. They're maintained at 37 degrees at a constant CO2 percentage to maintain the cells and keep them happy, actually. Here's an example of one of our cell lines that we use. Actually, it's a ROS1 cell line that we have derived from a patient.
Oops, got a glare. There we go.
And here's a microscopic view of this actual cell line.
This is how a ROS1 cell line looks under a microscope. You're actually looking at clumps of living ROS1 cancer cells. The little round spots are cell nuclei.
So we have the cells in culture, and once we need to do experiments, we transfer them to plates that are defined for the experiment that we need. In this case, I have a same cell line that I put it in these wells, and each plate was treated with different concentrations of 4 different TKIs. And I go from really high concentration to really low, and then once I treat it, I'm going to extract the proteins of the cells. So I put a solution that will dissolve the cells, and I'll collect that liquid to run and look for signaling proteins to see if the targets are being inhibited by the TKIs.
This graphic is called a Western blot. Researchers use it to detect protein signaling. This particular blot is looking at signaling from crizotinib-resistant ROS1 cell lines treated with different TKI combinations. Each set of colored bars represents a different concentration of a TKI or a combination of TKIs that have been applied to living ROS1 cells. If a ROS1 cell is signaling, it is not being inhibited. Green bars indicate the TKI is effectively inhibiting ROS1 cancer signaling, and therefore might be an effective ROS1 treatment. Lighter-colored bars indicate the TKI is not effectively inhibiting ROS1 cancer signaling and is therefore not an effective treatment. The 4 sets of bars on the far left show crizotinib-resistant ROS1 cells treated with crizotinib alone. The concentration of the TKI increases as you move from left to right. Not surprisingly, the first 3 bars on the left show typical doses of crizotinib are not inhibiting the cancer. However, the 4th set of bars from the left is green, which means that concentration of crizotinib is effectively inhibiting the cancer. Unfortunately, that concentration is too high to be tolerable in humans because it would kill healthy cells. Further to the right are bars representing TKI combinations. Some of the bars are green, which indicates those TKI combinations might work against crizotinib-resistant ROS1 cells. With additional research and funding, such experiments might eventually lead researchers to develop new ROS1 treatments that are both effective and tolerable. We hope you've enjoyed touring the Doebele Lab at the University of Colorado and learning about ROS1 cell lines and how researchers use them. Thank you to those who made this video possible.
Jenna had just introduced me. My name's An Lee, and I work for Dr. Bob Doebele. Actually, he's usually the one giving these types of presentation, and I'm usually the one back behind the lab benches doing the work. But in this case, we decided to switch it out a little bit and give you a different perspective on the work that we've done in collaboration with the ROS Wonders in generating new novel ROS1 cell lines. Next slide. These are just my disclosures and conflicts of interest. Next slide. So I'd like to start off with this pie chart of what we know about lung cancer. This is actually taken from the Longevity website, and this basically sums up our view of lung cancer as we know it in 2021. We see it as a collection of genetic alterations that goes awry and drives the proliferation of cancers. And why we think this is important is because if we're able to identify these genetic alterations, then there are FDA-approved targeted therapies that will be able to specifically treat a patient who has these genetic alterations and slow down their tumor growth. Now, the reason why I bring up 2021 is I like to flash back 10 years ago, back to 2011. Next slide, Jenna. Where, for the most part, back 10 years ago, we did not know much about what was driving the proliferation of lung cancers. It was largely unknown, as seen in this pie chart listed as other. The only other genetic alterations that were present that we knew of back in 2011 were mainly mutations in KRAS and EGFR. And then also around 2011, actually 2009, and then in 2011, there was discoveries of gene fusions in ALK and ROS1 that also contribute to the oncogenesis of lung cancer. Another reason why I like to bring up this pie chart back in 2011 is that, actually added the next slide. Back then, we were severely limited in the number of cell line models for us as researchers to investigate lung cancer back then. Almost like the pie chart in how it's broken up, the number of cancer cell line models were also limited in that we had a handful of KRAS and EGFR cell lines, but ALK and ROS1, well, basically there were only 2 ALK fusion cell lines that were available and only one ROS1 cell line. Now, another parallelism with 2011 is that was the year that I joined Doebele's lab. And both Bob and I came to the conclusion that if we are to truly investigate and interrogate the pathogenesis and the impact gene fusions had on driving the proliferation of lung cancers, we needed more cell line models, namely those in ROS1, ALK, and all the other ones. So next slide. And so we come back to 2011, and this is basically 10 years worth of work that we did in generating novel cell lines that are representative of lung cancer. And so we give them the name CUDO, and that's our designation for the cell lines that we created. It basically stands for CU, thoracic oncology, and we give them a number. And as you can see, over 10 years, we have accumulated quite a number of ALK, ROS, and RET fusion cell and NTRK fusion cell lines, as well as different mutations, insertions, and deletions in other genes such as EGFR, BRAF, KRAS, and MET. And sort of distinct and similar to that pie chart down there, the number of cell lines that are now available for us to work with is almost divvied up like that pie chart. However, one group of cell lines that seems to jump out at you as having several more representative models is the ROS1 fusion cell lines. And the reason for this is, and why I'm here to talk to you today, is because of our collaboration with the ROS Wonders. Now, next slide. So just a quick deviation. This is sort of our workflow as to how we're able to generate cell lines here at CU. Basically, a patient that comes through our university is seen by one of our clinicians. They're then consented under our rebiopsy protocol so that they give us consent to work on their biopsy sample. Once that is in agreement, on the day the procedure of where they have their procedure, whether it is a surgical resection, a core needle biopsy, or a draining of a pleural fluid, we are able to obtain a portion of that tissue sample, fresh, unfixed, and that is then brought back to our lab where we do some disaggregation, kind of break up the tissue, and then plate them out for expansion and isolation of cell lines. This is the part that takes the most time and varies from 6 months to a year. And after several rounds of expansion and isolation, and if we are successful, then we are able to generate a cell line, which we call it, and we give that CUDO designation. As you can see from this workflow, there are two places that are limiting our ability to make a cell line. First is during, the main obvious point here is in the expansion and isolation of actually deriving a cell line from a patient. Basically, to this day, we still are only able to have about a 30 to 40% success rate. The second limiting factor of generating novel cell lines here is actually, we are limited by the number of people who actually come through our the clinic here at CU. Now, one of the benefits of being able to collaborate with the ROS Wonders, it gave us a new way to obtain more samples. You click. So when we partnered with the ROS Wonders, what we were able to do is reach out to the patients, not in that they actually don't have to come through CU, but if they're able to donate either their fluid or or if we are able to does collaborate with their clinicians to have them send us fresh tissue, then we're able to collect more tissue samples, and that increases our chances of making new novel cell lines. And it is because of that, which resulted in such a high number of ROS1 cell lines that we were able to make here at CU. Next slide. This map of the states highlights all the 19 tissue samples that we were able to obtain over the past 4 years from ROS Wonders. And as you see, it's a diverse, we're getting it from diverse regions, and we're able to get some even from outside of the United States, namely Canada. Next slide. The end result of that is that as of right now, we have 12 ROS1 cell lines, of which 7 came from ROS Wonders. As you can see from this table of the ROS1 cell lines that we have available, they are diverse in the type of gene 5' fusion partners that are with the ROS1 gene rearrangement, although CD74 seems to be the most prevalent one. This chart also highlights a lot of the big data analysis that we were able to do, namely in part from the funding that ROS Wonders had generated. Namely, we were able to do, for most of them, RNA-seq analysis. So RNA-seq analysis is is a assay that we're able to do to ask what are being expressed at the RNA level from these cell lines. We were able to do whole exome sequencing, which allows us to interrogate all the types of mutations that undergo from these cells. We were able to do, for a few of them, global phosphoproteomics, whereby we're able to understand the different functions of various proteins in these cells and how certain signaling pathways in these cells get affected. And additionally, we've initiated some drug screen assays to test different compounds that may be effective in halting the proliferation of these cells. Next slide. Not only were we able to do analysis here in-house, we were able to now share out these ROS1 cell lines to other research labs throughout the nation who does basic science research. We also were able to give them out to various institutions where they actually needed these ROS1 cell lines as control samples to validate their clinical diagnostic tests in order to see if their tests are able to identify ROS1 fusion in a patient tumor. So that is quite significant. And also we were able to partner with other pharmaceuticals who needed these cell lines in order to test their novel compounds and drugs for the treatment of ROS1-driven lung cancer. Not only were we able to do stuff here in this, share out the cells here in the States, we're actually able to collaborate with folks in both Canada and also in Europe. And the reason why I want to highlight that is the two labs that we were able to collaborate and share our cell lines to in Europe, both in Netherlands and Belgium, are labs that has projects that are funded by the ROS Wonders. So in a way, it kind of all feeds back and helps, the funding helps in other labs that are being funded by ROS Wonders. Next one. This slide just kind of highlights some of the publications and scientific meetings where we were able to make use of the ROS1 cell lines. I'd just like to highlight the 3 publications that are currently out there. The first one was by Carolyn McCoach, who was here at CU, and she used the ROS1 cell lines along with the ALK cell lines to define the various types of resistance mechanisms to targeted therapies. And there are several mechanisms that we're able to use to do in vitro studies on the ROS1 to show that they are the mechanisms of resistance. The second paper was also here done at CU, and it was by Curtis Davies. And here it's actually a very interesting paper in that we were comparing the different types of methods to detect ROS1 gene rearrangements in lung tumor patients, or patients with lung tumor. And we show both advantages and shortcomings of whether it was a DNA genomic-based test versus an RNA-based assay. And then lastly, the paper from Dana Neal is from Trevor Bavona's lab. And here, they're over at UC San Francisco, and there they used the ROS1 cell lines to look at where different different fusion partners of ROS1 localized to in the cell. So like I had shown earlier, the different what we call front end or 5' end of the fusion partner, whether it's CD74 or SDC4 or other types of front end fusions, they may localize to different parts of the cell and therefore affect the way it signals. And then I think with that, that kind of concludes my portion of the presentation. Let me pass it back over to Janet.
Thank you, An. And thanks for sharing all of that hopeful information about the cell lines and the work that the ROS1ers have been able to do with the University of Colorado. I'm going to talk a little bit about how the ROS1 Cancer Model Project actually works in terms of getting the specimens to the universities. We actually have two places now that are doing these. One is the University of Colorado, and in this partnership, when the patient knows they have a procedure coming, then they contact the University of Colorado, who does a consent remotely. The University of Colorado then ships a collection kit to the patient. The patient has either their biopsy, a surgical resection, or a pleural fluid drain, and sends the specimen kit back to University of Colorado, who's provided a shipping label. University of Colorado then starts processing, and they try and do it hopefully within 24 hours of receiving the specimen, but more like certainly within 48. And if there are successful cell lines created, and as An said, it works about 35 to 40% of the time, then those cell lines are validated and characterized. The two places mainly where the ROS1ers participate are helping to contact patients and put them in touch with the University of Colorado, and then also money raised by the ROS1ers in conjunction with Alchemy, the Adario Lung Cancer Medical Institute, paid for the validation and characterization of the cells. This is actually a clinical trial listed on clinicaltrials.gov. We also have a partnership with pattern.org. You can go to pattern.org and register online at any time. And then when you know you're going to have a procedure, then you do the consents. And then pattern.org coordinates all of the specimen and data transfer with your healthcare provider so that the patient doesn't have to do anything. Both of these have some key features. It must have fresh tissue from a resection or a biopsy, or else fresh fluid from a pleural fusion drain to make the cell lines. The whole point is to capture live cancer cells and help them stay alive. But we only take specimens when you've already had a medical procedure scheduled. We don't ask you to have a procedure just to gather the tissues or the pleural fluid. And the bigger a surgical tissue sample you can give them, the better chance of success. But here's a key: you have to consent prior to the procedure. So as much advance notice as you can give us is wonderful. At least a week would be good, because you have to have time for the consent for them to ship you the packaging and have it shipped back. the focus here is on generating cell lines for research. We are not generating models that can then be tested to find out what treatment works for a patient. And the reason for that is because the testing that is used has to be done in a certain type of approved laboratory called a CLIA lab before you return results to patients. And not all of these tests are done in a CLIA lab. We protect the patient's privacy all the way through. The samples are de-identified so that nobody knows who they belong to. You can revoke your consent at any time, and there is no cost to the patient. And at the moment, the ROS Wonders are not having any expenses either. University of Colorado primarily funds their program by having pharma pay for the cell lines. And at the moment, pattern.org has grants that cover our work. When the cell lines go to pattern.org, at the moment, they're going to the Broad Institute, but we may also have the option in the future to work with MD Anderson. So that is the end of the presentation, formal part of the presentation. And now I'd like to have our panelists join us, and we will have a more open discussion. So welcome, Amy, and hello again, An.
Hello.
Hi. So we have some general topics to discuss. And please, in the audience, if you have any questions regarding what a cell line is, how it's used, how this program works, please put it in the chat or actually use the Q&A link, and we will bring them in.
And actually, I just want to interject because we already had one question that was brought up, and I think you answered it for the host and panelists, but it was whether or not this study was open to patients outside the US. And so since I don't think everyone saw your answer on if you want to clarify Colorado's policy about US versus ex-US.
So for us, the main part for us to obtain sample is to get the informed consent from a patient. And so we've been able to do remote consents. Well, all these are remote, but remote consents to some international parts of the country. I mean, we've done Canada, and I think we had the possibility of doing one in Australia, but that never came about, but and sometimes in Europe. So it is feasible. I mean, there's always, there's a little more hoops to come that gets to getting the samples here back to the US and whatnot, but in theory, and actually it's been feasible to be able to do, to obtain samples outside of the US border.
And I believe part of the difficulty with that is shipping, especially now with all of the problems with transportation. You have a much better chance of getting a successful cell line if it's processed very quickly. If there's delays, it gets really difficult. So once we get outside the US, it's much more difficult to get the transportation.
So it looks like Joyce has a question that really was kind of the thing that we were considering to kick off our discussion. And it's kind of what happens if you find something interesting with a cell line? So to what extent are these cell lines currently informing, practice, and how are they being utilized? So how are they supporting the development of new treatments? So maybe you can kick it off, and we can just give our listeners insights into the process and what your next steps are if you find something interesting as you're working with these cell lines.
So ultimately, with these cell lines, both Bob and I have the agreement that a lot of the research that we do, and we try to tell the patients up front, that it's to inform future research and to help inform future practice on how to overall treat ROS1 driven tumors, and so a lot of our research, a lot of the research we do is still basic science, and so we use these cell lines to help us inform and write papers and present at meetings a lot of our findings that these cell lines help us to inform. On some rare occasions, if we're able to make cell lines and generate them rapidly enough and glean some types of maybe resistance mechanisms, we do sometimes get back to the clinician treating the patient just to give them an idea as to what is going on with their tumor. Now, we always broach it with the fact that these, these are cell lines that may not be representative of the initial tumor sample that we received from them. And so it is always, we always suggest additional clinical testing for them to pursue in order to maybe alter the patient's therapeutic treatment plans.
Well, and I think more broadly, the point we want to reinforce too, and sorry, Janet, I didn't mean to cut you off, is beyond just the individual's treatment plan. It's broadly informing, research that can guide development of new treatments and new therapeutics. And, so I come to the conversation, one, having trained as a cancer scientist myself, having worked with cell lines in the laboratory, having worked with and created mouse models, but then also in a previous existence when I was in Georgia, I was focused on and helped build out a statewide biorepository. So our goal was to provide these resources to our cancer scientists, because we know that's often a rate limiting resource for driving new cancer discovery, is having access to these very precious samples, from patients. So I can't stress enough how important this effort is in advancing our understanding of cancer, of lung cancer, of, identifying new treatment opportunities. And so Janet, I'll hand it to you.
So I just wanted to build on that. You noticed on On's list, he listed the different fusions. ROS1 is a fusion that it swaps parts of the gene with other genes. And we know of, I think we're up to over 20 pairing partners. So the problem is, when crizotinib was developed, it was developed based on one mouse model. That's the cancer that existed in one particular person, and everybody's cancer is different. So as we get more of these models, we get them with different gene pairing partners, we get them with cells that behave differently, we get them that have developed resistance to certain drugs, and all of those things are things that the researchers can study to find out the biology of ROS1 and how it's behaving. If I remember properly, you told me that there are some cell lines that developed resistance to the drug the patient was on, but then there was one cell line that was inherently resistant to crizotinib from the get-go.
Yeah, and so that kind of represents the diversity of the cell lines that we were able to create, where sometimes it mimics a patient's resistance to our targeted therapies, and at times otherwise, or becomes totally refractory to any other additional therapies. And that's always, for us, it's very interesting, and that's when we love to collaborate with other researchers to, take a different view of how to get at this problem of resistance to targeted therapies. And the more models we have, the better we are to almost get a good landscape view of all the possible mechanisms there are. And so by not being limited to just one cell line, we are able to interrogate multiple cell lines with multiple different types of targeted therapies. And that helps us increase our understanding of the biology of resistance.
Well, one of those papers that was generated based using some of our cell lines was looking at these different types of resistance. Different oncogenes that drive cancer behave differently. So the people who have EGFR positive, the majority of them had one specific resistance mechanism that showed up, and a drug could be developed for that particular resistance mechanism. But for some reason in ROS1, at least half of us have some kind of mechanism that has nothing to do with the ROS1 gene. It's something else in the cell that changes. And we don't know what that is. Without these cell lines, we can't figure it out.
Also, to make a note of that, it is interesting that you say that, because so there are, as we know with targeted therapies, there could be on-target resistance in that ROS1 cell line, or the ROS1 tumors will develop a mutation in the ROS1 kinase domain that now blocks the drug from binding. Now the cell lines that we've created from the ROS1 patients, none of them actually has any of these kinase domain mutations. So they are more bypass or different co-occurring alterations that is driving the mechanism, which makes it very interesting and very interesting to investigate what are these bypass mechanisms.
And I believe that Dr. Doebele presented a couple of years ago that by looking at our cell lines and others that you have developed, he found that there were some resistance mechanisms that were common to different cancers, like RET and MET and EGFR and ALK and ROS1 all had the same thing, the same pathway in the cell go awry that was causing the resistance.
Correct. And we do now starting to see these prevailing themes or prevailing pathways that seems to be the fallback for these cancers to use when they are deprived or blocked by their primary oncogenes. And that's why it was really valuable to initially partner with the ROS1ers to generate all these cell lines. But I could easily see us partnering with other, let's say the RET, with RET fusion patient advocate group to generate more RET cell lines, since that seems for us to be a limited factor in how we're able to look at those mechanisms. And so I see our partnership as a model for others to work with.
I was actually curious when you presented your list of all the different cell lines you have created as part of your repository. Each one is assigned a number. And so I was wondering if there's a correlation between, the number, like I noticed there was a KRAS, which was your very first one, and then ROS1 was number 2, NTRK was number 3. So do you prioritize cell lines on kind of when these different biomarkers become actionable or relevant, or do you just take things as they come in anticipation of perhaps they will be informative and useful? So can you talk us through kind of that? Do you take anything that kind of comes through the door, or are you prioritizing certain biomarkers now more strategically?
Yeah, so actually early on, basically what started this project, it was actually a side project for our lab because we didn't actually have real true funding to develop new cell lines. So we couldn't take this big broad approach to take every patient samples that came through the CU Oncology Center and try to make cell lines, because it was basically just Bob's lab and me doing the cell line derivation. So we did prioritize the tissue samples we were going after. We already knew we had representative cell lines for EGFR and KRAS, but back then, given that there was only one ROS1 and 2 ALK, and then soon after that was a RET fusion, we started targeting just rare gene fusion patients with either rare gene fusions. But we also targeted, because the population had a lot of these rearrangements, were actually in the never smoker population of lung cancer. And so so for all the unknowns, we were really targeting the never smokers population.
So Amy, you want to talk a little bit about the process for developing a drug? Say they find a particular TKI that is effective in dealing with resistance in the cell line. What would be the steps that would happen to see that eventually become a clinical trial for a new drug?
Well, I mean, we were certainly talking about that, and An has a valuable perspective on this as well. The conversation typically, the process that we think about in getting, something from the bench to the bedside involves multiple steps. So there's kind of this discovery phase of, what we describe as in vitro experiments. So perhaps that's working in cell lines. Typically then you're going to take some of those findings and expand that to animal models. So often we talk about there's kind of this coupling of multiple different types of models for cancer drug discovery. So you've got cell lines, you may have, we talked about organoids, which kind of preserve some of the 3D components of a tumor kind of in a dish. Or then you have animal models. So we also talk about PDX, patient-derived xenografts, or as you talked about, sometimes we can take a piece of the tumor and implant it under the skin in the mouse model. Those are also useful. So, we think about expanding, but we know that's also a source of limitation for some of these rare mutations and fusions, right? So we have our own challenges there. And then eventually you'll have something that can move into a phase 1, clinical trial in patients. So, there's multiple steps involved. And I do want to kind of just back up and revisit the conversation though about you mentioned that in creating these cell lines, it's often a 6 month to a 1 year process, perhaps. And, I hear this kind of feedback from patients a lot because we all appreciate the urgency that comes with trying to find new solutions and new treatments for patients with cancer, especially with stage 4 lung cancer. Trying to manage those expectations, and trying to move science as fast as we can while still, adhering to certain protocols and certain processes. So I think we want to maybe talk a little bit about that and what you do once you get that tissue, you start creating a cell line, then there's characterization and validation of that cell line before it can really be used in a meaningful way. So let's chat about that a little bit.
Yes, for us, of course, we'll just stick with the ROS1 patient cells. When we actually have a true cell line that's growing out, like as Janet showed in the flash that we believe to be driven by ROS1, then we do some initial validation. Of course, we have to show genetically that there is a gene rearrangement in ROS1 and that this gene rearrangement is present and is also being expressed in those cells. Now, the second part for us as basic researcher is to then use various types of assays to see how responsive they are to ROS1 tyrosine kinase inhibitors. Because for the most part, when we receive these samples, these are coming from ROS1 patients that has already been diagnosed with ROS1 and that are currently on ongoing therapies to treat their ROS1-driven tumors. And so there's a chance that most likely that these cells would not be responsive to the primary drug that they on. And so we do those assays where we test to see if these cells are able to proliferate in the therapies that the patients are on. We extend that to all other types of ROS1 inhibitors that are available to us, just to get an assessment as to the level of responsiveness the cell line that we created is to how the spectrum of their response to different ROS1 TKIs. We also look at the signaling pathways that are involved with that's involved with driving these cell lines. Sometimes we'll look at some other pathways that we know are involved given our past experience. So we'll interrogate those pathways. And then, as I pointed out on that table, we've done big data analysis where we do RNA-seq to look at expression, we do whole exomes to look at mutations, and proteomics to look at protein phosphorylation. Then that is our validation and establishment and assessment of the cell lines. And then they become available to both, not only to the researchers here at CU, but to other researchers looking to look at ROS1 driven lung cancer in other parts of the nation and the world.
I just wanted to add in addition to Ann's response about whether you could accept tissue from or samples from outside the U.S., Barbara Van Heer at patent.org responded, and at the moment they are limited to samples from the U.S., but they're hoping to add Canada sometime early next year. So So there's another question here. So if somebody's having a surgery or a rebiopsy, they're assuming the tumor has to be large enough to provide tissue for both. We would expect you to have your doctor take as much tissue as he needs to make good medical decisions for you. And if there's something left over, you can send it to the University of Colorado.
Right. We always prioritize because each clinician has their own pathology clinical validation that they need to do the assessment on either the biopsy or their surgical resection. So what we get should always be secondary to the initial assessment done over their clinician.
Okay, sounds good. So what are some of your primary challenges in making a successful cell line?
Well,
in 10 words or less.
Let me make, because I mean, there's always variability in the type of samples that we get. And over the years, we found actually that getting patient pleural fluid has been the most successful means of generating a cell line, actually more rapidly than, let's say, a surgical resection or a tissue biopsy. That's one. There are still several unknown factors in generating a cell line. We still don't know why we only have a 30 to 40% success rate. There's also, presence, when we get these tissue samples, there's also presence of normal cells that actually has the ability to grow because the cancer cells may influence the growth of normal stromal, what we call stromal cells. And sometimes that overtakes the culture and we're not able to isolate tumor cells from stromal cells. And so that kind of influences our ability to make cells with specific genetic alterations. It's still a learning process, I guess.
Yeah. I mean, I think it's important for the audience to really appreciate how much of an art form this is. And, when An says he joined Dr. Doebele's lab a decade ago, he brought a wealth of expertise and experience to the table and then has really, increased that over the last decade. So in developing each of these cell lines, they have very specific requirements. And so that may be the media that they grow in, that may be the CO2 level that they are maintained at. And so it's all, a bit of trial and error to figure out what are the appropriate conditions that each one grows at. And it can be a little bit customized for each cell line. So, it is a bit of, an elegant effort that An and his team have established to create these cells.
Well, and there's the additional complication, if I remember correctly, that these cell lines sometimes, for no particular reason, will decide to die.
Yeah, no, we definitely have had times where we think we have a good cell line expanding, and it just poops out. It just doesn't. It stops, and it's disappointing. But we try to capture as early as possible and kind of freeze away some of the early, what we call early passage cells, so that if we can freeze them away and then down the road, if they start to die off, then we come back to the initial passage in hopes of trying to expand those.
I see a note here from Upal. We just want to reiterate that when we generate a cell line, this is not something that gets shared back and given to the patient's doctor to work with. This is just shared within research labs. So if you're hoping to use that for your own research, this is something we do to try and help additional ROS1 patients in the future and learn more about the disease.
And help inform the entire research community on lung cancer.
Well, so just to reiterate what you had on one of your charts, these have been used to validate tests for detecting ROS1. They've been used to study with different cell lines from other diseases. They've been used to look at resistance mechanisms. And I know it was also shared with Ignited, the people who developed the drug that's now approved called entrectinib. So there's a lot of benefit in having these cell lines. So here's a question from people. What can we as ROS Wonders do to help get more information out about these cell lines and the importance of getting the consent done before surgery? That's been a challenge. One of the things we ran into early is it seems like everyone in the world schedules biopsies on Fridays.
Yeah.
Which means that An had to be in the lab on Saturday to receive the package.
Although we did have one case where I actually was able to arrange for FedEx to deliver it to the tissue sample to my house, which I then had to run back to the university to process over the weekend.
Wow. So
because FedEx does not deliver to our university on the weekends.
Well, I believe both of our protocols allow you to sign up at any time, but we usually will not consent until you know that you've got a surgery. And Barbara Van Heer, if you can correct me if that's different on pattern. So in terms of trying to get the word out about these, I mean, we're putting information on our website. We've helped people in the group, and enough people have been doing it in the group that people are starting to ask us saying, hey, I've got a procedure coming up, let us know. All we ask is that give us as many days notice as you can. If you'd like, tell us I've got a procedure the next day, there won't be much we can do.
Right, because ultimately for us, we still have to coordinate with the clinician who's actually doing the procedure. And each institution has their own protocols and limitations on how they're able to share out the sample. So it always, not only is a dialogue with the patient, but the dialogue also is with the clinicians who's doing the procedure.
So we're getting close to the top of the hour. One thing we haven't asked is, on if people want to access these cell lines for research, what should they do?
So the way we have it arranged is that they can, for researchers, they can easily email or contact both Bob Doebele or I, and that gets the ball rolling, because then we can initiate what we call a materials transfer agreement, where it's an agreement with our institution, since we hold onto the cell lines, and their institution, and reach an agreement as to how the cell lines will be used. And then once everything is in place, then we ship the cells to the basic science research labs.
So, Amy, is there anything you want to say in conclusion? I will give on a chance.
I mean, I think, the key points are that we want to stress to the listeners is the value of participating in this project and the importance of having access to these cell lines for advancing cancer research. As I said, I trained as a basic cancer scientist, and when I was doing my postdoc at Vanderbilt, routinely worked with cell lines that were established, but there's we have kind of different cell lines. So we have some that have been maintained for years and years and years. And then we talk about primary cell lines, which are derived directly from patients and create these cell lines that we're talking about today. And there's, so much value to having those, because we can appreciate some of the, I guess, nuances of the individual patient's cancer and how it responds and reacts to different treatments. So it's a tremendous thing that you all, the ROS Wonders, are doing for the cancer research community and establishing this partnership with Colorado University. We're very appreciative to An and to Dr. Doebele for their work to establish these, because it's making a tremendous difference in understanding the disease, accelerating new therapeutics, and giving hope to all the patients who are dealing with ROS1 driven lung cancer and other mutations as well. So I would encourage everyone who's listening to, find out more and explore whether or not you have the opportunity to participate. So if you're newly diagnosed or at the point of progression, have that conversation with your doctor and your care team so they know your wishes. And if you are, thinking about participating, then we encourage you to do that and to lay the groundwork so that when that time comes, it's kind of ready to go.
And just for me to reemphasize that, I am truly grateful that to Janet for reaching out to our lab and starting this partnership with the ROS Wonders, because it was only through that we were able to generate these unique ROS1 cell lines. And I hope it serves as a model for other patient advocacy group that they can reach out to us and help increase the number of other cell lines that are available for other types of oncogenes.
Thank you to the Doebele Lab at the University of Colorado for their partnership and collaboration on this project, as well as our other collaborators, Alchemy, Pattern.org, and Longevity.